I wanted to go and start talking about the adaptive immune system. Yes.
Real quick, is the lab lecture schedule, is that going to be updated for what we're doing today? This says 17 and 18 for today.
No, because then I have to back up there.
Oh, okay.
Yeah.
So are we shifted like?
We might be,
Like,
yeah,
okay.
we might be shifted a little bit different, so.
But the exams and the quizzes are still.
Oh, they're going to be, if you put in there 16, you could, well, okay, for that reason, I can see why I should maybe update that. I will, because we were probably pretty well set when we took the last quiz. So, yeah, I'll redo it so that you'll know. Because I think the next exam is like a week from Wednesday or something, or a week from today.
Yeah.
Something like that. Yeah, so I'll update it so that you'll see the ones. But it'll be from whatever chapters we covered until now. Okay, so on this particular chapter, we had talked about the innate immune system before, and we kept alluding to the adaptive immune system. And when we looked at all those blood cells, we looked at some lymphocytes, and we looked for the B cells and T cells. Those are all the ones that we're going to talk about now. And I'm going to have to always be up here because I have to change this. And so maybe we can even just start with how this looks up here. Let me see. It's not working. Oh, I put this in. Sorry. Oh, so this does work a little better. Not great, but okay. So if you look at this this organism right here, you can see where the PAMPs are. Do you remember what PAMPs stand for? What's the first word? Pathogen. Associated. Molecular. Patterns. So these are things like, and now ignore this part with the antibiotic. So this is like the pili, this is like the generic, maybe flagella, that could be something on the surface of the organism. And these are the things that your innate immune system, they recognize it. There's just patterns on all bacteria that they can recognize. And so they're indiscriminate. If there's a pathogen in, they don't care whether they understand it's a pathogen, maybe it could be even one of your cells, they will just attack it. So it's indiscriminate, but at least it's very quick, and what they're hoping to do is keep the pathogen at bay. But now we're going to start being much more specific. So you notice, even though we have a flagella here that would have a toll-like receptor that recognized it, we also have individual kind of antigen determinants. These are maybe proteins that make a flagella, and that flagella will have a very specific protein that makes part of the flagella of a very specific organism. So while the flagella in total can be recognized by PAMPs, a very specific one right here would recognize that particular one and would activate your adaptive immune system. And the same thing on the pili. They have very certain regions on the pili that will be recognized by your adaptive immune system. So it might look like it's doing double duty, but the basis of having your innate immunity is to try to recognize any pathogen and then hold it at bay or kill it, and then take the sample of it up to your adaptive immune system and see if we can get longer-term reactions. All right, so this is really, we're going to focus on the adaptive immune system, and we'll kind of look here. We looked at this before, so we can see up here, this is, right now you should know where the B cells and T cells are, and where they're located, it looks like at the top here, but on the bottom here, but it's just that candy cane shape. So up here where the B cells are, and we really call that the cortex. So this part right here is the cortex, and that part down there is the medulla, and that's where the T cells are. So B cells are on the top portion of the cortex, and the medulla is where the T cells are. And the lymph and the blood is flowing through here, bringing samples of all kinds of things that are in the fluids of the body. And what they're hoping is that the B cells and the T cells out here that recognize a specific epitope on one of those pathogens, they will be activated, and then they'll start giving you long-term memory. So remember, here's the lymph nodes. We have many, many, many lymph nodes. So as the blood is flowing through, or the macrophages are displaying as they come to the lymph nodes, there are many, many lymph nodes for it to have to go through. And that means to show the proper B cell or T cell that has that epitope, it has to go through a lot. And it could be days before it actually finds it. All right, so just so you kind of remember this, you would not have to draw the entire structure, but you would probably need to either draw or recognize this structure in here. So being able to draw it or label it, that kind of depth. Okay, so I guess this isn't going to be perfect. Okay, so this is the bottom of the previous slide. So this is just telling about the B cells and T cells. And that germinal center there is where the propagation of the B cells will be, and the plasma cells, which don't make much sense to us right now, and the antibodies. Let me try to straighten this out a little bit. I don't think it's going to work very well, but I'll try each one. All right, so this is our next slide, and really what's happening is since we have two kinds of immune system, an innate immune system and an adaptive immune system, there has to be a big difference between them. And the biggest difference is that your adaptive immune system takes a different approach. They don't just hit everything. They only hit things that are very specific to what they recognize. And right now that doesn't make much sense, because you think, how do they recognize it? We're going to find out that there's about 10 to the 14th, that's like a million trillion, different antibody receptors and T cell receptors, and they're looking for their epitope, which means that any pathogen in the world, you already have antibodies and T cells against. It's just finding when you're infected with whatever that organism is, it's just finding your T cells and your B cells so you can activate them. Okay, so the approach that your adaptive immune system takes is four-pronged. So they want to have something that's very specific. Remember, they're not looking for PAMPs. They're not looking for general things. They're looking for very specific for each pathogen. So each antibody or each T cell will look at only one epitope. They're not going to be looking at a whole bunch. And then also tolerance of self. We really don't have that with your innate immunity. They just start killing. It could actually kill some of your own cells. But if it just doesn't look right, they're going to start to kill it. And they want to have very minimal self-damage. So your adaptive immune system, if they're very specific and they're on target, they're only going to cause a inflammation and a problem with that particular pathogen. It's not going to be something that's going to be throughout the body. And then immunological memory. This is a really big thing, because once you are infected with a pathogen, your adaptive immune system, if it kicks in, it will be able to make antibodies or have cytotoxic T cells made, and those types of cells will have long-term memory. So they will stay in your body for basically years. It could be even for 10 or 15 years. If it's something that, it depends on how often your body has seen it, so they will make strong antibody reaction against it. So this immunological memory is something that's really important to have. So antigens. This is a term that we use when we say that there's something on the surface that is going to trigger a reaction by an antibody. So it's an antigenic reaction. It's not a PAMP. It is something that is very, very specific to that organism. And we can see, like on, this is coronavirus. We had this portion right here, the receptors that we have are kind of our spike proteins that we had out here. These are the ones that triggered the immune system. So they took a vaccine and they made an antibody against these proteins on the outside, so that that antibody could cover these. So if you think of an antibody like a little tent, covering all the spike proteins. And if the spike proteins were covered, they were unable then to bind to the cell that they had to get in, and so this coronavirus that got into the body was totally useless. It could not get into the cells that it wanted to. So antibodies are against a very specific target, and here we made antibodies against a specific target that would keep the coronavirus from being able to infect. So the antigen is kind of a general term. They call almost everything that causes an antigenic response, and that means your adaptive immune system will respond to it. And epitopes, they're made with either proteins, that's probably the most powerful one. They could be made with sugars, carbohydrates, and we'll see on the next slide we have carbohydrates, and they can even be made of DNA. So the last two I mentioned are not very strong because they get degraded pretty fast, so they don't last long enough for your adaptive immune system to recognize it. But proteins do. Okay, so for kind of the take home from this is these four special cases of an adaptive immune system versus your innate immune system. Okay. All right, so now we're going to take each of those strategies that adaptive immune system has, and we're going to kind of break it down and talk about it more. So we have those four defensive strategies that it took, and one of them is the specificity. And the specificity means that if they have a pathogen like this that enters, and let's say they have another pathogen over here that enters, unless it has these specific epitopes on it, your adaptive immune system is going to ignore it. It only is going to attack a pathogen that it has the epitope that matches with the T cells or the B cells. And again, about 8 or 9 slides from now, we'll talk more about T cells and B cells, so this will make a little bit more sense. So specificity, your B cells and your T cells, these are white blood cells. They are going to recognize very specific epitopes on a pathogen, so they will only attack that particular pathogen. And the epitopes are going to be made up of protein, I mentioned, or carbohydrates or nucleic acids. Proteins are the longest lasting. Remember, proteins are made up of amino acids. Those amino acids fold into a very specific shape. They have lots of intermolecular interactions that keep that shape the way it is. So that allows your antibodies or your T cells to be able to recognize that particular shape. And that's what it's making antibodies, or that's what it's acting against when it's defending your body against a pathogen. So these regions are on the cell. They're like 3D fingerprints, and they're recognized then by your adaptive immune system. They are not the PAPs that we've talked about before. Even though they could be in the same region, they could be on a flagella. Every flagella is going to be specific for each pathogen. They'll have a lot of general proteins in there, and that's what your innate immunity is looking at, but they'll also have specific ones for each pathogen. So if it's made up of proteins, it's much more potent because carbohydrates and nucleic acids, they break down a lot faster, and it's harder for your adaptive immune system to recognize it. And immune deficiency, this is really when your body doesn't have the ability to recognize specific antigen. So if they don't have antigens that it can recognize, then your body is unable to elicit an immune response. So this immune deficiency is it's really quite devastating to the body because you don't have your second line of defense. Your first line is your innate immunity, but if you don't have your adaptive immunity, usually these pathogens can overcome the body. And I mentioned we had 10 to the 14th different epitopes that your B cells and your T cells already today have. So you think, why would we have so many epitopes when there's probably not that many? Well, there must be that many in the world, but there's not that many that we encounter. So at birth, your body goes through a recombination of your variable regions on your cells for what they're going to bind to, and so those are the ones that you have for the rest of your life. And they are enough to defend you against any pathogen that you will ever see. So again, let's kind of Sorry, 10 to the 14 trillion, that's a lot of them. Let me see. So for this one, our second strategy, well, first strategy was your adaptive immune system is much more specific. The second strategy is they want to have tolerance of self. So they don't ever want to be able to make antibodies or cytotoxic T cells against one of your own cells. So they have to have a way to be able to recognize an epitope that is your own epitope and should not be attacked. And there are things called regulatory T cells. They come from the thymus, and the new T cells are generated in the thymus. And what is happened for your cytotoxic T cells, or your helper T cells, both of those, they are challenged by your regulatory T cells. They're shown epitopes that are your own cell epitopes. And if your T cells, your helper T cells or your cytotoxic T cells, if they respond to those epitopes, they are destroyed. And so this keeps your body safe from having a T cell start to recognize your own cells as a pathogen. So this is the idea of keeping tolerance of self. So we're making sure that the T cells are very tolerant of the cells that we have our own. And these T cells, these helper T cells, not the regulatory T cells, but the helper T cells, they need to, because now they've been trained, they need to take any epitope that your B cells, which make antibodies, any of your B cells, if they want to make an antibody against a specific epitope, it has to be approved, because they could make antibodies against any cell epitope if they had not been screened out. So what happens is a good screening is to have the helper T cell that has already been screened for all of these self epitopes. Whenever a B cell wants to make antibodies, it must go to a T cell and it must be activated. So the T cell that already recognizes what epitopes should have antibodies made against it, if the B cell is saying, I want to make an antibody against this epitope and displays it, your T cells will destroy that B cell if it is showing a self cell, okay, or a self epitope. So the idea of the B cells, if they need to be destroyed if they are going to start making antibodies against self. Okay, so this is when they do, and things happen, they go awry in the body all the time. This is when autoimmune disorders occur, when you start making antibodies against your own proteins. And when that happens, it just, sometimes it's not deadly, but it just causes difficulties. It causes joint problems, itching, just uncomfortable spots, different things. So these autoimmune systems, lupus is one of them, and some other ones you might have immune responses against things like penicillin. So all of this are ones that are really harmless, and yet your body does try to make antibodies against them. And these are, hapten is a word that means it's something that should not have immunogenic response, but it does. And usually what happens is things like penicillin, we should not have an immune response against penicillin. But when penicillin or even poison ivy gets into the body, it binds with another protein, poison ivy another protein that's on the skin, and penicillin is another protein, and I'm not sure exactly where it is in the body, but it binds to that, and it causes now an immune response to that bound protein. Yes?
For autoimmune disorders to happen, is the cause of them the T cells aren't trained in the thymus anymore, or is it unknown?
No, they don't always know. If they knew, they probably would be able to kind of have some kind of a deterrent for it. But I think there's a lot of different things that cause autoimmune disease. If your immune system, and it could be even your basophils and things that we're not going to talk about for allergic reactions, they can just go awry, and that's really not controlled by your T cells or your B cells. So there's lots of things that cause it. So I don't know if we could just tag it to the T or B cells. Yes?
Doesn't the thymus disappear at the age of puberty?
I don't think so. Yeah. I'm not aware if it is.
I think it does, yeah.
Okay.
So, like, that's why.
Is it to shrink down or is it go away?
I think it goes away. Don't quote me on that, but.
Okay, well, yeah, I'll look it up too, but, and so what would be the question then if the thymus?
So like where are the T cells like moving
Yeah,
after that?
yeah, I would wonder. And I mean, your body is making, you have to have your T cells and they're replenished all the time. So I don't know, but maybe it just shrinks to a much smaller size, or I'm not sure.
Shrinks around puberty, early 20s.
Yeah, the shrinks or disappears.
Shrinks and eventually disappears over time.
Okay.
So it loses its ability to produce new T cells.
So that doesn't seem good for your body, huh? Not to ever produce new B cells or T cells. Okay, so anyway, let's go to the next strategy. So this is our strategy number 3, and this we've probably read about and heard about. We know about COVID. Some of the problems with COVID was that your body made an overzealous response to having a virus within your lungs, and so those overzealous response was cytokines being sent out from the cells, and that was bringing more and more immune cells into the lungs, and every time that happens, they're going through blood vessels and they're leaking some of the fluid, and your lungs would get more and more fluid, so inflammation, and there was just a what they call a cytokine storm, lots of signaling going on, calling lots of more molecules to that location, and just causing more havoc. And this was also seen in the 1918 Spanish flu pandemic. They couldn't understand why many very young people, there were soldiers and stuff in the Spanish-American War, and they found so many of them were dying, and they were dying of the flu. And that was unusual because they were usually healthier and older people did not die. And they decided what it was is because their immune system was so strong that it caused these over responses, and that's what caused the pneumonia, and pneumonia is what usually killed them. Okay? So something called a cytokine storm is a storm of cytokines that your immune cell is sending out more and more signaling molecules to try to bring more immune cells to that location to be able to handle whatever problem it is. So this is something that your adaptive immune system tries to avoid. They tried to be very specific and they tried to be very controlled. This is usually something that happens because your innate immune system is trying its hardest to keep the pathogen at bay. So it calls more and more innate cells to that location to try to kill the pathogen. And they feel the more of them, the better. But in an adaptive immune response, that's not the case. They have very, very specific responses. So this just tells you a lot of people, 50 to 100 million died from Spanish American flu, and then the 7.6 was just with COVID, and this is, they really stopped taking data now on COVID. So this is another defense. So this, remember, there were 4 defenses. And this defense is looking at the memory. They once thought that memory was a sign of an advanced organism. We have memory in our cells, but they also find now that cockroaches have memory. They can get infected with something and they can get memory cells and they can fight against it. Bacteria also have a memory system, and we're going to see that at the very end of these slides. So they kind of abandoned that thing, saying that these are higher order organisms that have it. We think that most organisms now have some way to recognize pathogens that they have been infected with before so that they can avoid them. So immunological memory just is a way that your body makes a response. You can see here it takes a little bit of time right here to be able to respond, and this is IgM. That's one of your immunoglobulins. It is a antibody, and it's just called IgM. We'll see on a couple slides a little bit later, they're shaped like, there's got a 5 pentamer, and they're really, looking for sugars. So that is why they have 5 of them there, because the sugars are a little bit less able for them to detect. So the hallmark is the first thing that responds to a pathogen that comes in. Remember, your innate immune system has gone to each of the lymph nodes that you have, displaying on its surface MHC class II molecule, displaying the pathogen that they have discovered. And your B cells and your T cells that are in the lymph nodes are each looking at that pathogen. And they're either saying, yes, I recognize that pathogen and I can make antibodies against it, and your T cells say, yes, I can recognize that also. Or it's saying, I've never seen that before. So they just pass on it. So your macrophage or whatever it's displaying is going from lymph node to lymph node to lymph node. And finally, it will find a response. One of the antibodies will eventually recognize it, and then it will start making IgM. That's the first one. It's a pentamer, so it can grab a lot of pathogens with it. It's got 5 different binding spots, but it still takes a long time. And then once it gets to a certain level, by that time your IgG, which is your primary antibody, your IgG starts to kick in. So this is what most of your antibodies are made in this kind of format, but your IgM is the first one. So when you're taking blood samples and you test and you have a lot of IgM, that probably means you very recently have gotten the disease. You might still have some IgG, but not a lot, and that just means your body is really starting to kick in and make an immune response. And then the next time, the second time, because now you have memory, and your IgM and your IgG, they are already established for that particular pathogen, and as soon as that pathogen enters the body again, they will make an almost immediate response. So this you can see it enters, and then almost immediately your IgG is really high. And that means you're expressing antibodies that will be able to tag that pathogen and keep it at bay, either keep it from infecting cells or tagging it so that your adaptive your innate immune system can come in and then chew it all up. So they're just keeping it like it does for SARS. It just binds onto the spike protein, and it won't let the spike protein get into a cell. So as it's bound to the spike protein, that gives the innate immune system time to get to that virus and then chew it up and get rid of it. So the adaptive immune system can't kill. All it can do is attach to and tag a particular pathogen. Okay, so memory is one of the strategies that are very important for antibodies and the strategies for the adaptive immune system. So now let's talk a little bit more, I think this gets all on the slide, talk about exactly what makes up the adaptive immune system. So we know that it's the B cells and the T cells, and those T cells have cytotoxic T cells, they have other kinds of T cells. B cells only have B cells, but B cells turn into other things that eventually make antibodies. So these are in the humoral part of the body, which means it's all the fluids. So your antibodies are flowing around in the liquid parts of the cell or in the lymph. And these are where they're searching all the time. Once they have recognized their epitope, then they are searching in your body all the time. And of course they wane over time. If you see your pathogen today, and your body starts making antibodies, and you see it again in 2 more days, and in 5 more days, and in 8 more days, you are making antibody after antibody after antibody. But after a while, maybe you don't see it again for a month, and maybe it's 2 months, and maybe it's a year. You still have, if you have memory, you still have your body circulating with these antibodies that are still against that pathogen that you haven't seen for 5 years. And if you see it again, it will make not a really fast response, but a pretty quick response. It takes it a while then to notify and start making more antibodies against it. But if you've recently seen that pathogen, you may not even know that you've been infected with it again because they'll have so many antibodies there to dampen down that response that your body will not respond to having a pathogen in it. So this is what a B cell would look like, and this, having a flat surface, there's 100,000 of all these identical receptors on the B cell. These receptors are the ones that are going to recognize its epitope. So you can see these, and I'll show you another slide. There's a whole different structure that might be on the cell. But the cell is just covered with the kind of receptor that is going to recognize its epitope. These are proteins that are making this epitope, and they'll fold it into a certain way, and then they'll stay as receptors to try to identify its epitope. Okay, all right. So this is a B cell. It's the first component of your adaptive immune system. And now let's look at a T cell. So the T cell looks relatively the same. Its receptor is a little bit different. These are called glycoproteins, so it's still made out of proteins, but there's also sugars involved. And these glycoproteins do exactly the same thing. They try to bind the epitope of whatever pathogen they are made for. So again, we have just trillions and trillions of these T cells and B cells waiting. They're naive, they've never seen their pathogen before, and they're waiting to be activated. So again, in this, we have, I mentioned the T helper cells, and I said what the T helper cells do is they will activate the B cells, because the T helper cells have been trained on what is self and what isn't, and B cells have not. We also have something called a cytotoxic T cell, because remember, B cells are in the fluids of the body. So they are going through the body constantly looking for epitopes, trying to match maybe a new pathogen that has come in, but they cannot see inside a cell. So if a pathogen gets into a cell and it's starting to kill the cell, cells will often take what they go in through endocytosis, they will chew it up and they will display it on their surface. So your normal body cells are displaying on their surface pathogens that they are infected with. So they have to have some kind of a cell that can come around and look at the cell and kind of look inside it by looking at what's displayed on the surface. So if they can recognize these cytotoxic T cells, if it matches their epitope, they are going to latch onto that cell, and then they are going to secrete materials into the cell to kill the cell. So that's like a cell-to-cell combat, and that's something antibodies cannot do. So they only recognize a soluble pathogen that is floating around. And these can actually recognize something that's inside one of our cells. So they have totally different functions. So now T helper cells, cytotoxic T cells, and then also regulatory T cells. There are many different kinds of T cells, but regulatory T cells. Okay, so questions about this one or a connection between anything else? So, I don't know why this isn't advancing forward. Okay, so let's look at, because I said we have trillions of these. How do we get trillions? So when we have these B cells and T cells, when they recognize, you have 100 trillion of them, 10 to the 14th, they're all naive until they actually recognize their epitope. Let me see if the slide is totally on there yet. So they don't really clone themselves, they don't make more of themselves until their epitope is recognized in the body. So many of them just are going to remain naive the entire time. You'll have antibodies in your, not antibodies, but B cells and T cells in your lymph nodes that are never activated. So your immune system, it can just about any epitope that ever comes in, they'll be able to recognize it. And remember, they're looking at something very specific, and it's going to, the T cell and the B cell is going to recognize the same kind of epitopes. The B and the T cells, they're going to eventually clone themselves. Once they have been activated with their epitope, they're going to take it in, and when that happens, they start generating new T cells or new B cells. And those T cells are going to be T cells that are able to recognize one specific epitope. So we have what we call effector cells, and these effector cells are the ones that are going to be able to target the pathogen. Now, the B cells help generate antibodies, and so it's not the cell itself that does any damage, it's the antibodies that are produced. And when those antibodies produce, they're soluble, they leave, they go out through the bloodstream, and it's those antibodies that bind to the epitope that the B cell has recognized. So it's not the cell itself, it's the antibodies that's produced. But the T cells will actually, when they're activated, they are cytotoxic, some T cells, and they will do cell-to-cell combat. And then the effector cells are the ones that are going to target the pathogens, either the antibodies or the cytotoxic T cells. But you also have these memory cells. So these memory cells can wane if it doesn't seem it's pathogen for a while, and that could be like for a year or 2 years or 5 years. But when it does see it again, it will kick in and start making new cells of itself. And that doesn't take long. That doesn't take any approval of any other cell to do that. It's already got the approval. You already have the memory cells. It doesn't take very long for them to kick in again and start making a whole bunch of cells that can be effector cells and then get rid of the pathogen. Okay, so it can be reactivated pretty quickly on short notice, but eventually they do wane, and sometimes we get boosters if we get our immune system targeted by maybe vaccines. So here's another kind of thing on the same topic. So this could be a B cell, and this is a B cell, and this B cell is showing one of the ways that it gets activated. I said another way that it can get activated is the macrophage encountered a pathogen. It took that pathogen in, it chopped it all up, it displayed on its surface on an MHC class II molecule, it displayed on its surface a piece of the pathogen. And then it went through your blood system and your lymph, and it showed that piece of the pathogen to all kinds of B cells and T cells in your lymph nodes. So that was one way. And once that is shown to a T cell or a B cell, that will also activate it. But there's another one where your pathogens are still in your fluids of the body, and they are being filtered through your lymph nodes all the time. So your pathogen itself, you could have an antigen that's on a pathogen that is actually going to activate, and this is going to be a B cell. So here's a B cell. This B cell doesn't know self from non-self. So this B cell, it takes a antigen, and maybe it saw it in the bloodstream, so it takes it in. It actually takes it in through phagocytosis, and now it puts it into the interior and then displays it on the surface. So this is actually displaying on an MHC class II. You can kind of see it down here. It's displaying this a piece of that pathogen that it recognized with its own receptor. It says it matches my receptor. I have the ability to be able to bind to this. So it then wants to make more of itself, but it doesn't. Well, I mean, it can't do that without approval. So this slide kind of connects to this other slide. So here, let me go up here. You can see up here you have a lot of different receptors on the B cells. So here came the epitope in, and this was the only cell that matched the epitope. All the other B cells within that cortex of the lymph nodes didn't recognize it, so they're still just sitting there. They're waiting for their epitope to come by. Okay, and this one matched. And so now this B cell is the one who has taken that in and is displaying it on its surface. So again, the B cells have not been trained what is self and what is non-self. So what will happen is it will have to go to a T cell. Here's a T cell. This is a helper T cell, and it has a TH2, there's a 2 helper cell, and you can see on it, it actually has a CD4 on it. So do you remember what CD4 was? Does that trigger any memories?
Receptor for HIV.
Yes. And so this is one of the ones, HIV, when it needs to get in a cell, that's what it's looking for. And so unfortunately, it has to be one of our T cells. Not one of them, I mean many of our T cells have the CD4. So if it has a CD4 on it, HIV is going to infect that cell, and this is the very cell that should be recognizing a pathogen and alerting the body, and it is now being defeated. So again, that's one of the reasons HIV is so devastating. And what it does is when it gets in, remember what HIV does? It integrates into the genome. So this T cell may not be making virus, but it's got that HIV in its genome. So every time this T cell is replicating and making new T cells, it's making new HIV. And then at one point in time, and they don't know what always triggers it, but HIV then will come out of the genome, out of being a provirus, and become viral. And when that happens, it usually attacks more and more T cells. That makes your T cell count go down, and it makes the HIV count come up. So either you get into full-blown AIDS from that, or maybe your immune system is strong enough that it kicks in and it can fight it. But it only can do that so long. So that's why we have so many drugs that will help assist a cell get rid of the, or dampen down the ability for the HIV to propagate. Okay, so anyway, what we have now is we have a B cell that has found an epitope that it matches. Could have been a self one. So it takes it over to a T cell and it says, can I make antibodies? And so the T cell will say yes by secreting interleukin-4. Okay, so this is a cytokine, and what it does is a signaling molecule that says, yes, I'm going to activate you. You may now make more antibodies. So this cell, now you can see the B cell, it starts making more of the B cells. You can see it's a B cell, it's got its receptors on it. Those receptors are the exact ones that match the epitope that it just matched. So now we have a whole bunch of them, but the B cell is not really going to do anything. What the B cell will do is after it's cloned a whole bunch of them, it will start dividing up, and some of them will remain as memory cells, so they look just like the old B cell. It's got the same receptors on it, so that if you're infected with this particular pathogen again, those receptors that recognize that epitope are going to be available to recognize that pathogen and then come back and start cloning again. And when it clones, one of the other things that clones is these plasma cells. So you can see here's our original B cell. They set aside some for memory cells, and then the rest of them went to make these plasma cells. On the plasma cells, there are no receptors. The only thing that a plasma cell does is it has the genetic material within it to make the antibodies that have the epitope recognizing ability that's going to be on their on the antibody, and I'll show you what an antibody looks like in a second. So all of this happens is the plasma cells make the antibodies. The B cells don't make antibodies, and I say that kind of at the beginning because they are connected together, but in reality what you should say is the plasma cells, which come from the B cells, are the ones that make the antibodies. Okay, and then of course the B cells also set aside some memory cells so that your body will be able to fight this very quickly the next time it is engaged. Okay, does that make sense? So now we have kind of a series of molecules, and here's that word again, cytokine, that just means a signal into the cytoplasm, and a good CD4 should be a molecule that you recognize, and then these are just little side bars that you can read. So here's the structure of an antibody, and I put on here kind of what's important. These are antibodies or products of the plasma cells, so don't say that they're a product of the B cell. The B cell is the originator, but the plasma cell is the thing that actually makes them. This is what an antibody looks like. It's kind of a stylized form. This is really what the antibody looks like as far as a protein. But these are all proteins. There's 4 proteins in here. There's a light chain here, and these heavy chains. And you can see these have coloring to them. This part up here in green for each of them. These are the epitopes where they recognize the pathogen. So these are going to be very specific to each pathogen that they bind. This stuff down here is called the constant region. So an antibody is formed the same. It has this big region down at the bottom, and that is going to be used it's called an opsonin, and so what it is used for is it's to attract macrophages to the site where an antibody is bound to, and so that tail sticks up, and it's signaling to the macrophages as, hey, come over here and get rid of, chew up whatever I'm binding to, because this is a pathogen. So it just kind of helps keep the pathogen stationary or at bay or unable to infect a cell, and then it gives time for the macrophages to come over and then gulp that up and then chop it all in pieces and get rid of it. So this tail part needs to be recognized by all macrophages. It can't be specific for a pathogen. So the tail part is called constant, and it has kind of a constant structure to it, so it can be recognized. So again, antibodies cannot kill. They have no cytoplasmic granules or something like that. They are just protein structure, and they bind onto their epitope, and they just hold on, trying to keep that epitope from being used. Okay, so these, again, they're in the fluids. This is really what the more stylized one looks like. And remember, they're made from plasma cells. So I want to talk more about these antibodies, but for this particular slide, you should focus on this shape up here. This is the one, if you were to draw it, not this curly-Q one, but this one right here would be the one that you would want to focus on drawing and being able to identify the proteins that are on there. So now I said there was 10 to the 14th, which is, I mean, if you just start writing 10 and all the zeros, that's a huge, huge, huge amount of different antibodies. So you think, how can you have a genetic material in your body to be able to produce something like 10 to the 14th different things? It's like impossible. So they said there's got to be a way that these antibodies are produced all randomly like this in a way that's efficient for the cell to do. So what they realized then after doing a lot of research is they have these regions. They have this variable region up here in green, and then they have this yellow region, and then they have these J and these C regions. So it gives, these are all different, and you can see there's 300 of them, so it gives a great variety for a cell when it is going to decide to make an antibody or a receptor for an antibody. They go, oh, I think I'll take one of these, and then I'll take one of these, and one of these, and then of course the constant region. So the next one says, well, I think I'll take this one down here, and I'll take that one right there, and I'll take this. So the combination, if you take 300 times 50 times 4, etc., it just makes up to an astronomical amount. And you also have, this is the heavy chain, this is the light chain. So you have even this, 300 times 5, etc., to show the variations that you can make. So with all that variability, you do come up to something like 10 to the 14th different variations of every antibody that we have. And it's made from all one genetic component within the cell. It just skips around and takes different portions of that and then just puts them together. And once that's done, then that's done for that particular cell. It does not make more, because each cell is only going to have one antibody that's made. It doesn't make a whole bunch. Okay, so this is how they do them. So it's not that you have to memorize, each of these, but just kind of know that there's a variable. The genetic portion is variable in certain regions, and each region is taken, once, and then they can have a lot of different varieties. Again, you don't have to know there's 300 and 500. Just know that there's quite a few to choose from, and it chooses from something different. Each antibody is different because of the choice that was made by the B cell. Okay, but each B cell does not make any more than just that one antibody. Even though it has 100,000 receptors on the surface, they're all exactly the same receptor. Okay, does this kind of make sense? And the region that's most variable is this part up here. So you can see where the variability is coming up here, and that part is all right there. And that's, if you go back here, you can see this is the part that's binding the epitope. And here's the part right here that's binding the epitope. So we mentioned before, and each antibody, it's called the immunoglobulin, and we have 5 different ones. And I mentioned IgM. So IgM is the first one that comes out. It's a pentamer. And this is, I'm going to have I'm only going to say some of this stuff, and then you should really go through the next 2 slides a little bit more in depth. But the IgG is the second one that's made. It's the major one that we're going to have in circulation almost all the time. The IgM is the one that's first, and it's a pentamer. I have a better slide that I'm going to show, so I'm not really going to go on with this anymore, but this has a little bit more words to it. This is a slide that's a little bit easier to focus on, although it's not really very dark. But I think because we see what they look like on top here, by looking at that, you can see what the shape of it is. So here you have the IgG, you can see it's just kind of a little V. Here's the pentamer, and then here's a dimer, those are IgA. So IgA, the half-life is very short. It activates the complement, or it doesn't activate the complement, and for crossing the placenta, it does not. So really the IgG is the only one that does. And this gives you a little bit more information about the antibodies themselves. Okay. So any question about these? So these are the antibodies that the B cells are going to make. And they can make, and all of these antibodies will recognize the exact same epitope. They just have a different way of binding and different places that they bind. Okay. And so this is just kind of a review now that you see we have things like memory cells. So at the beginning, I think, this kind of goes off the slide, but at the very beginning you have that primary response, and you can see it takes days, and then the IgM is the first, and then the IgG kind of kicks in and a little bit higher, and then it wanes off. And what happens is, because these are all just made out of proteins, eventually in your body all your proteins are rotated all the time. So some proteins maybe last a week or a month, some of them last only hours, some of them last just days. Your body doesn't want to make a protein and then have the protein live forever, because maybe you don't need it. And we can just chop that up and use the amino acids and stuff to make something else. So the proteins, they're always going to be destroyed after a certain period of time, and of course your body, if it's exposed to the pathogen again, it's going to make new antibodies to help the system. And then again, if you're exposed to it, this is only a couple of days, and look at how high your IgGs go. So you still have the IgM come in. It comes in about the same time, and then it really starts waning off, and the IgG kicks in. So this is just helps the body fight that infection almost immediately. So allowing your body, we're going to talk about vaccines and stuff, not today, but in another lecture. Getting a vaccine and gives your body exposure to something right away, so you make antibodies and you do some of these things, but sometimes you're not exposed to it long enough, so you don't make long-term memory. So even though you have a vaccine, your body, you're going to have to get booster shots for that vaccine, and not all vaccines, because some vaccines have live organisms in, or at least attenuated organisms. But your body has to see the organism over and over again before it can make a strong response. If you just show it once and then not again, your body says, well, whatever this was, it's not going to be a threat. But the body has to see it several times. Okay, so here we are. It makes a really good, strong response after being exposed to it. This is the second time. Okay, so let's look at this part. So these are antibody interactions, and I'm not going to go through, I am going to go through all of them, but I'll let you read them on this slide to see what we're going to be talking about. So instead of just reading off the slide, we'll actually go through them. So this first one, let's see, antibodies binding epitopes to mediate phagocytosis. So here, is this one up here? Yeah, here is where phagocytosis is occurring. So here's a pathogen, here's a macrophage, and it is destroying a pathogen that has been tagged with the antibodies. So we can look at each of these. The end result is going to be where your macrophage comes in. So I like to ask a question that says, antibodies can't kill. Why are they effective? What can they do that it makes them effective? Because they can't kill a cell. They have nothing to kill. So what is it that it's doing that is making it so valuable? So you can see the antibodies right here, right? The antibodies are actually encountering a pathogen, they're binding to that pathogen, and they're making sure the pathogen can do nothing else. So this pathogen might be a virus, and now it's got all these antibodies on it. It would not be able to get into one of your cells. So these antibodies are not killing it. It's just keeping it so that the virus or whatever it is cannot get into your cells and cause you harm. And because it has these constant regions of the constant regions of the antibody pointing out, this is where your macrophage is going to come in and say, hang on, I'm on my way. I'll come there, and what I'll do is I'll engulf this, I'll chop it all up, and you'll be done. It gets rid of all the antibodies too, but you have zillions of antibodies. So the antibodies are just holding on to this to make sure a macrophage can come and then dispose of it. Does that make sense? So no ability to kill, but it just keeps whatever it is at bay. So here is a way we call this neutralization. This we call viral inhibition. We're inhibiting that virus from getting into cells. Here's neutralization. So here's, maybe we have this pathogen. This could be even like a snake protein or a snake bite protein. And it gets into the cells, and then you're given some kind of antitoxin, so you get these antibodies. And what they're trying to do is take this protein from being able to get into one of your cells or attach to one of your blood cells. So if you give the antibodies quick enough, if this is floating around in the bloodstream, this is going to be tagged to be eventually chewed up and gotten rid of, but at least it can't do its job. It can't get into one of your cells and cause harm. So this is just called neutralization. And then we have opsonization. So here is a bacteria, let's say, and it's just tagging it. It's just sticking on the surface, it's recognizing some kind of epitopes on it, and it's sticking on the surface to say, hey, macrophage, come in and get rid of this. So opsonization is just a way to tag something to be destroyed by macrophages or your innate immunity. And then here is agglutination. So what this can do is just take lots of pathogens and get them together, and they just become a big clump. If this was in a solution, and we were doing a test to see if you had this pathogen, and you did an antibody test, and you put the antibodies in, and all of a sudden you had a big clump down in the bottom of the petri dish or something, or maybe some fluorescence happened in the bottom of the petri dish. That's really what this is doing. Agglutination just means you're clumping things together. And antibodies can do that because, remember, antibodies have two different arms, so we can bind two different ones at once. Here's one binding here and here. So maybe we should think about taking a break. I'm going to quickly look and see how many slides, so you can decide then if you want to take a break or just barrel through that. So let's get through this one, and then I'll look and see the additional slides. All right, so this one we can see agglutination is just means they're going to bind these all together. And of course, this would also stop the pathogen from being able to do anything else. And here, precipitation is the same conceptual idea. Agglutination and precipitation is where you are you're clumping them together so that you can either see them in a higher concentration or definitely keep them together for a macrophage to come. So again, all of these, the question, like I said, I ask a lot is, if antibodies can't kill anything, what good are they? I mean, what do they actually do? And we can see here they do an awful lot. And this region down here, we call the constant region that I said on the antibody right here, that part that sticks up and stuff, that is a constant region. And it's, we looked at the somatic reorganization, we saw that that part does not get revamped. It usually stays just about the same for all antibodies. And now we can see why, because we want a macrophage to be able to come no matter which antibody it is, it wants to be able to recognize that tail, that fixed region. So another mistake that I see sometimes is I'll ask you to maybe mimic one of these things that we've put on the board here, and you'll have, let's say it's just a bacteria, and here's a flagella, and so you'll have the antibody like this. Okay, so this will be the fixed region binding to the pathogen. You want like the epitope on here, so you want this region with the tail sticking up. So you wouldn't want the tail part. I guess this could look also like one arm of the antibody, but I'll try to make it more distinct. This is really the tail. So you want to make it like this, where these are the variable regions that are binding to the organism. Okay, so just don't do it with the tail region, because this wouldn't help at all. The macrophage cannot recognize this region, it can only recognize this. Okay. All right, so I wanted to go through here quickly and see the slides. I think some of you have these. You can see the number of slides. So there's probably about maybe 15 or something, so we probably should just take kind of a short break and then come back and finish these up. Is that maybe a little ring or walk around or something? And I'm going to, before we do that, so it looks like the only one I have is gone is so 1, 2, 3, 4, 5, 6, 8, 9, 10. So Andrea would be gone. Is that the only one? Andrea? Pardon me? What'd you say? Is there some, say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So we have 13, 14, 15. So Andrea is one of the ones that's gone. And are you thinking that she might have dropped? Is that what you're saying? Or? Pardon me? No, you didn't say anything. Okay. So then I'm not sure who else. Who else can you look at and say is gone? Oh, yes, okay. Oh, and, okay, you're here. And so else at the table, this table, and then Chris is here. And so, oh, John, where's John? Oh, so sorry, John. Okay. See, I haven't seen that group there. Okay, so that's that table. So there's one more person missing. Who is it? Oh, okay. Oh, that might be now. Yep, you're right. Okay. So really just two. Okay, so we'll look kind of now and see what the time is. 7:37, so maybe 7:47. Oh yeah, okay. So when I reviewed them for other people, this putting them directly underneath doesn't give us the feeling that these are the ones that are eliminated. So I don't know if you can, because it's very clear what you've done here. Whether you could turn it a different way and then be able to put some on the sides and have always one side be eliminated. Yeah, because I mean, it's and I like this where you kind of give us a trail to go by a color, so that's really good, and everything is cleanly typed. A lot of people have number 1, 2, 3, 5 or something like that. It just calls it, this is just straightforward. The only thing is, it's just not really understood that everything underneath is menu cop. So if you usually put it to one side, that does it. Yeah, okay, yeah. But it still can be directly underneath it. Yeah, it has to be. Oh, well, I mean, you do have it kind of to the side. So this kind of is like one side. That might work though.
Yeah.
Most of the ones that we had, they had wide names out here.
I want the.
Or out on the side over here. Yours is like.
So
So I know that you're saying this is negative, so negative goes back. And then you're. And I can't tell you whether it's right or not, but I can just tell you that. But yeah, just work with you.
This is, I don't know.
She's from.
Like that, so
Yeah.
so major.
No, I already said what I was gonna do, in case you hadn't brought it.
This is something.
Yeah, and whether there was anything wrong with it or not, that doesn't inhibit the point value, you still get the same point.
It's just a long.
It's just that you had it done.
Priscilla's recording.
Oh, and the other thing, we have lab on tomorrow, right? Yeah, and so it's the food lab. So I was going to bring the lab so that we could kind of look at it. They do suggest that you bring food. And really what there is there's some reagents that we put on the food and we mash it up and we put it in a machine and it gums it all up and then we test it. So I think somebody said pizza they would bring or something. I don't know with all the cheese or something if it was that will gum up the system to do it. But you should really bring different foods and then if it works, it works. If it doesn't, it doesn't. They'll bring a whole bunch of things. They have blueberries and cucumbers and celery or things like that. I mean, kind of uninteresting things. But I would like cereal or some things that I kind of wonder what they're good. But they're just looking for the kind of microorganisms that will be in those foods. Yep. But you don't have to bring it. Last time they had quite a few things for us to test.
She's a weirdo. She's black hair, glasses.
Black, brown.
She makes you feel
I like that.
Is there a pull a picture of her, dude. And every muscle.
Did they highlight the ones that are more community?
Spectacle, I literally failed. I got 25 out of 50 times. No,
Mine's always to have it this weekend, next weekend, instead of the other time. Yeah.
It's either going to be the bestest or it's going to be something stupid. Yeah, I know. It's no. She gives us, I think, 5 extra points on I know. I just, everything she updates, she gives us the list of like help us, like learn, and she doesn't have 16 credits. And how do we supposed to ever figure out most of you all and you all
I'm running away up to show you. It was 3:15.
There's one that we have that
And that's not a maximum. Should we do it?
I'm very prepared. She was like the girl that went out tonight. I need to be up on the
And that's not a maximum. That's just your regular.
Well, I could have 4.
4 different.
At 32 roll down. So what did she say about the quiz or the exam? Is she pushing you back or is she?
No, I think just the
So whatever we
lecture.
covered.
Yes, like
So
whatever
the
we
4
covered.
chapters.
Yeah, it looks like for the exam.
So the last 3 are just gonna be on the final exam.
Okay, so are we ready maybe to re-engage with this? Yeah, yes.
What did you say about the exam? So it's next week, are we just gonna have the 4 chapters then? The 8?
Whatever's from the previous thing, I haven't even looked at it to set it up. So
But it'll
I
still be
don't
next
know.
week.
Pardon me?
But it'll still be next week?
It's wherever it's scheduled, yeah. Yeah, they like it to keep it scheduled because all of the classes are doing the same thing.
Okay.
Yeah. So, I mean, next time it's just too many tests. Yeah, it takes up time. I think because other people just do it online. Yeah, so they do it on Friday or something like that. Here, take this test on Friday from this time to this time is, you know, I don't know, but that's not my style. So I can just, I don't know how you can know if somebody knows something if you do it all multiple choice and it's just randomized. I don't know. Anyway, yeah, I'm doing it this way because they want to keep the classes as short as possible.
Will you put that information up?
Pardon me?
Will you put that info up?
What info?
What the
Study
chapter,
guide?
the exam?
Well, I'll do the study guide like I always do.
Because it's not on either yet.
No, it isn't at all. No. I mean, I'll get to it when I can. Yeah, I'll get to it when I can. I mean, I'll do it, it's probably, let me see, so this is Wednesdays, so Thursdays, impossible. So it's probably gonna be like the weekend, so it's gonna be just before, like 4 or 5 days before the exam. That's as much as I can do, yeah.
So the last 3 chapters are just gonna be on the final exam?
I have no idea. I have no idea. Is there another exam after that? I haven't looked.
That's it. We only have 3 exams, right? And then the final?
Yes, the final is cumulative of everything.
Yeah.
Yeah. Yes.
How far are you along to grading the blue books?
Oh, so the blue books are pretty close, but because this is finals week at Fullerton, so probably next week I'll be totally done with them, and it'll be a matter of days. Then I'm
Oh, okay, so then we'll have it in before the end of the
semester?
Yeah, semester.
Oh, yes, definitely, definitely.
I was like lost words now.
It's like the end. Yeah, oh yeah. I thought I would try to prioritize it before like quizzes and stuff like that, but then all your stuff started. Yeah, it was so easy to grade a quiz to give you 20, 40 points. And then to keep, then only do one book, and I think now you're still waiting on more stuff. So at least this gives you a little bit of feedback constantly.
Yeah, no. I was only curious because I wanted to know if you were planning on updating it at that very end, like just uploading it then, so that's what I was asking.
It won't be at the very end because it seems like, I still can make progress every weekend, there's something on it, and this next one, because we have final exams, probably won't get much, but the week after that, then there's nothing. I don't even have to go to school on Monday, Wednesday, or Friday. Their school. Yeah, so once they're done, then it's just like oodles of time.
They're done with finals?
Well, there'll be no, this is the last week, like they had their ACS exams, so at least my chemistry courses did. And then the next week we have final exams. And then it's the grading of the final exams.
Do you like teaching chemistry?
Yeah, well, I usually teach biochemistry or nutrition or something like that, but chemistry is a very easy subject to do because it's all pattern forming. And so you put the equation down and you either get the equation right or you don't. There's no, I know, but, the grams I had wrong and I forgot to put moles and I didn't do this. So they know right away that this is wrong. Or anything that's verbal, you go, well, I meant this, so I put it in that order, but I meant it to be this. And so you can kind of see that it could be this, but so you don't know how many points to give that when somebody else was perfect. So it's really hard to grade anything different. So yeah, if grading-wise, I like chemistry, and I can get a lot of units from chemistry because I can grade that stuff pretty fast. Okay, so let's get on to this. So I want to give you some hints on this. These next quizzes and stuff that are going to be on these lectures, there's going to be a lot of drawing. There's going to be a lot of stuff that I mentioned that are on each slide. So you should be really focusing on some of the things I talk about. There's not much you can do on this other than I can maybe ask you to draw a particular interaction and kind of go along with the sentence of why antibodies are valuable. And this really shows why. Okay, so anyway, let's go on to the next part. This is, okay, so remember we talked about complement. So complement was a series of proteins. There's about 30 different proteins, and when those proteins do, they'll gather together and they'll make actually a whole. So when they make a whole in the cell, you can see here are some antibodies, and those antibodies are close enough that their tails up here will attract the complement. And these complement molecules will corral around those tails, and they'll make a little cluster, and then that cluster just comes off one by one, and they go into the membrane of the pathogen. And so the membrane then gets a hole in it, and of course everything inside comes out and everything outside goes in. So that will kill the cell. But these antibodies are really important because otherwise complement could get one of your own cells. There's really not much that complement has on it. There's no little receptors for them to be able to tell what cell it is. So if they have antibodies and the antibody concentration is relatively high, then they will be able to come and hold on to the tails, make this complex, and then the complex then can filter over and actually make the hole in the pathogen. So it is, and this doesn't really give you the illusion that there's a lot of antibodies there, but if there were only one antibody on this particular pathogen, the membrane of this pathogen, the complement wouldn't form because there's not enough tails for it to form on. And that would give us the security of, if let's say an antibody by mistake bound to one of our self cells, and if it only took one antibody tail to attract the complement, and then the complement then would filter over and make a hole in one of our own cells, that wouldn't be good. So this way, having at least several different antibodies in close proximity will allow the complement to form, and then it can just jump off there and make a hole. And we call this a membrane attack complex. This is another thing that antibodies contribute to. They provide the fixed region that the complement can attach to and then kill the cell. So you could say maybe this is a way that antibodies could help kill the cell. They could get together on a pathogen and then complement, but it's still the complement that's doing it. Antibody is not killing itself. It has nothing to be able to kill it. Okay, so this is a membrane attack complex. So this could be something I ask you to draw or give me an illustration of how complement and antibodies work together. So there's a lot in this chapter and in the previous chapter that could be asked more than just multiple choice. So more of a kind of show and tell kind of a question. All right, so this is, now we're getting in, the thing that we had before is we were talking just about antibodies. And antibodies are in the soluble portion of the body, and they're going from place to place to the blood system, through the lymph, and they're doing what they can to keep pathogens at bay. But this isn't going to help us if we have a pathogen that has already attacked a cell, and the cell is suffering, and we need to either get rid of that cell or bring attention to it. So these cell-mediated responses, let me see if I can get this. I guess this is good enough. I'll move it out. So cell-mediated, remember these are the cytotoxic, it's like a TC, so it's cytotoxic T cell. So T cells now are the ones that can do hand-to-hand combat with another cell. And this is the humoral response. This is where antibodies are all in the fluids, but when the cell is actually infected itself, antibodies will not do any good. It cannot see the epitopes when the epitope is already inside of the organism. So that means we have to have some kind of cell-mediated exchange between an infected cell and a cell that is non-infected. And so we have a couple of types here. So we have cytotoxic T cells and we have helper T cells. So the helper T cells are going to help us activate other cells, and including other T cells, to be able to become active. Because remember, the helper T cells are the ones that know what epitopes are allowed. And then the cytotoxic T cells, they also have to be approved by T helper cells to tell them that they can now go out and attack anything that they see with this epitope on it. So both of them they do peptide fragments. This is really what we've been looking at each time. You see the epitopes are going to be the peptides. Remember, why aren't carbohydrates and lipids very good epitopes for us to make antibodies against? They kind of just get destroyed pretty quickly. So if you're trying to make antibodies against something and you activate a epitope, and then the epitope can't find it again because it's gone or it's disfigured in a different way. When you have a protein that folds, the only way the protein is valid is if it folds in a very specific way. And so proteins almost always fold exactly the way they should, or they denature and then we don't worry about them. But a protein structure is much more antigenic. Yes?
Can you explain what it means, like soluble antibodies, like T cells, effectively that?
So when a cell is infected, one of your body cells is infected with an organism, that means the organism went inside the cell and is infecting the cell. Mm-hmm. So when you have an antibody that is soluble, that's out in the cytoplasm, it's going around, the cell that's infected has no way to tell the antibody, I have something inside me. Because the antibody only is looking at that variable region, and they're trying to find the epitope using its variable region.
Okay, so but are antibodies in the blood?
Yes, but the cell has something inside the cell. The cytoplasm and the membrane is around it. The antibody can't see that cell, and it can't break open the membrane and get inside the cell. It's only when that cell would display maybe an epitope on its surface that an antibody might recognize it with its variable region. But then again, all it's going to do is tag it and wait for a phagocyte to come on and eat that. But this way we have helper T cells that actually will secrete granules that will attack that cell and kill it. So these are actually cells that can kill, these cytotoxic T cells. So we'll see what their job is. Okay, so antibodies really are not effective once something gets inside. And then these two ones, they're going to recognize peptide fragments. And they go through the same kind of recombination to get a whole bunch of different variable regions. Let me see. All right, so here we have how do T cells recognize non-self. So we have these T cell receptors, that's what this stands for, and co-receptors that are going to be on cells. And it has to have an MHC on it, on a cell. Every one of your body cells, let me see if I can move this up. Down here's a little chart. Here. You can see this part. So this little chart says what's on particular kind of cells. So all nucleated cells have what they call MHC class 1 on their surface. Those are one of those little housekeeping genes. Remember the natural killer cell when it was looking for cells that were maybe had virus infected or cancer or something. They didn't have good housekeeping and they didn't put their receptors up on the surface. So these are one of the ones that they're looking for, these MHC class proteins. So all nucleated cells in the body except red blood cells have an MHC class 1. And macrophages, dendritic cells, and B cells, they have an MHC class 2. Those are the only ones that do. So the ones we've been talking about, like say the macrophages with their MHC class 2 are displaying, that is this protein right here. So your T cells recognize that receptor, that MHC class 1 or 2 as the receptor, and that is on your self cell. And then it's recognizing whether you have CD8 or CD4 on the helper T cell that help connect them two together. Okay? Okay. Oh, you guys okay? Good. Okay. All right, so these you need to know that the nucleated cells, all of them are going to have the MHC class 1, and the macrophages, dendritic cells, and B cells are going to have MHC class 2. So the MHC proteins are very unique for every human being. Well, they used to call them human leukocyte antigens. I don't think they really use that term anymore. And what these do is these mark body cells as self cells. So the body knows that that particular cell belongs in that tissue or in that body. And there's a whole bunch of different genes that encode these to make them all variable between individuals, but we're not concerned about that. So the idea, the thing I want you to know out of here is that we have an MHC class 1. All cells have that except red blood cells, or all nucleated cells, I should say. And then your macrophages also have MHC class 2 on it. So you could have an MHC class 1 and you could have an MHC class 2. So these T cells, they have a co-receptor on them, and now they have many other co-receptors, but the ones that we need to understand are CD8, and that's on a cytotoxic T cell. And then on the CD4 are the helper cell, T helper cell 1 and T helper cell 2. So that CD4, that is the one that HIV uses to help it infect cells. So it doesn't do all T cells, it just does it can do other cells. Because CD8, it also can be used, but CD4 is the primary one. Okay, so this just shows us that this is a way that your T cells can recognize self from non-self. All right, now cell-mediated, there's a lot on this slide. So cell-mediated means that we're talking about T cells now. And cell-mediated means that it is going to actually, the T cell has to interact with a body cell. So the T cell has to be activated. It has to see its epitope. It has to be activated, and then it can go around, and once it's activated, it can go then into the bloodstream, and it can go out into near all the cells, and it could just be looking for the epitope displayed on a cell that is really asking for help. It's saying, I've ingested this, I'm displaying it on my surface, on my MHC class 1, because I'm just a normal body cell. I'm displaying it on my surface because this, whatever this is inside me is killing me. And so I want you to know that, and so I want you to kill me so that I don't destroy the whole tissue by remaining here. So that's kind of the story that they're probably telling each other. So the first thing that has to happen is well, let me go down this side first. So we have the T helper cells, and remember they're recognizing MHC class 2, and these are CD4 co-receptors. So here's a cell. And it's going to be recognized, and the two things that it can be is you can be the cell-mediated receptor over here, the process that we go over here, and these are the processes that you'll have, and these include natural killer cells, and you have your B cells. So we've done this before. We've talked about the T helper 2 cell, and it has interleukin 4, and it stimulates the B cell to go ahead and divide. So we've talked about that side before, but we haven't talked about the same kind of T cell. Here's a T helper 2, but a T helper 1 is going to be stimulated by the same epitope. So this T helper 1 now is the one that's going to be activating the T cells, the cytotoxic T cells. So we kind of went down this side already once, and now we're going to go down this side. So the next slide is going to talk about this. So the T helper 1 has recognized a epitope, and it is now going to activate other cells. So I've mentioned a whole bunch of times about CD4 and HIV, so make sure you put that in your thoughts. So up here, you can see this is kind of what happens when the antigen, these are antigen-presenting cells, macrophages, dendritic cells, so they just kind of put a generic term on it. So this is a cell that's engulfed a pathogen. Here's a pathogen, it's broken all up inside, and then it's put onto the surface. These are MHC class 2, because this is a phagocytic cell, and so here's an antigen that it's presenting. And so here's a CD4 naive T cell that has come and looked at this epitope and said, yes, I recognize that epitope, so now I can activate other cells. So this one has now become activated, and they do interleukin 2. So these are T cells, and they activate themselves by interleukin 2. And we had interleukin 4 when we were activating the B cells. So here they're secreting interleukin 2 to tell the T cells, okay, make more T cells, set aside some of you to be memory cells, and then the rest of you are going to be able to go out and use the approval of the epitope that you are you can connect with. You can now kill the cells that you connect with. Okay, so that is where we've gotten our cytotoxic T cells activated. Or we really got our T helper 1 cells activated, I should say that. It hasn't gone yet to the... So here they are now, or down on this part, the T helper 1s are all activated. And so they're going to go all to these other cells to activate them. And that carries us over to this side. So now the cytotoxic T cells are going to be activated, and they have a CD8 receptor on them. And then they're going to go to like a virus or a tumor-infected cell, whatever cell that they're got an epitope to. They are going to go to that cell, and let's say this is a cell that's sick or something, and it's got this displayed on its surface. And then what's going to happen is this cytotoxic T lymphocyte is going to actually attack the cell and kill it. So here's our infected cell. Here's our connection with our cytotoxic T cell. It's got granules in it. It secretes those granules. They go in and they kill the cell. Okay, so you wouldn't want a cytotoxic T cell to be activated against some cells because if the epitope is there, they will actually just kill it because it's been approved for being eliminated. Okay, so that's why they have to be induced. They have the CD8 on their surface. They kill non-self cells, virus-infected cells, tumor cells, anything that is displaying on its surface something that it doesn't, that has been approved for killing it. Okay, so this is our T cells. So our T cells are going to do what we call combat with a cell. It's going to connect with the cell, and it'll kill the cell because the cell is displaying on its surface what kind of epitope is inside, and that is a pathogenic epitope, and so the epitope means that it needs to be killed. Okay, so then our next scenario, let's see, turn here. Okay, this is just the process of it killing. You get the epitope in, and it's displayed on the surface. I think the same thing that we had kind of talked about before. So this would just be a T cell, kind of a small little picture I showed you before. And then this is kind of a concept map. The concept map tells you different ways that your adaptive immune system is working. This is antigen presenting cell. And so any cell that can present an antigen on it is going to be these kind of cells. So that usually activates some other kind of cell. Here's a naive T cell, let's say. That naive T cell is going to then become a helper cell. And that helper cell then can turn into memory cells, or it can activate on T helper 2 cells. It can do B cells. It can be T helper 1 cell, and it can do cytotoxic T cells. So T helper 2 goes to B cells, T helper 1 will go to cytotoxic T cells or other T cells. Okay? And this is just a way, and of course they all want to go down to memory. If it's a B cell, it goes down to memory, and that makes plasma cells, and we make antibodies. So it's just kind of a concept map. You can kind of look at it. There's a lot of other independent stuff on here for this one. I'm not using this this textbook for this class. So there's a whole bunch of other stuff that is on this text that you don't have in your text. So we're just really stopping at this point. Okay. So let's do this. I think we've almost done this exact slide before. This is kind of a wrap up. And I do this because this will probably be important on the final exam. It pulls together a lot of the immune system and disease, etc. So why is HIV so harmful and difficult to treat? And we have talked about this before, and I think we've mentioned it. It infects CD4 receptors. It uses that to help infect. And these are mainly really important cells in your body. So here you have a pathogen that can actually infect one of the cells that are supposed to be our safeguards of our body, and that is going to have devastating results. And humoral immunity is adversely affected because if we don't have T cells to activate the antibodies, then we don't have that liquid antibodies to count on. So B cells aren't activated, T cells aren't activated. You have really no immune system left if HIV is infecting. So low T cell count, low cell-mediated immunity, low B cell, severely weakened immune system, and mainly it's almost death is coming in at program time. So that's why we have so many, or have spent so much money, and hopefully we can continue to do that on HIV research and HIV prevention, because it is extremely effective. And once it is, the only result is going to be death over a period of time. Okay, so this is HIV. And the other thing I wanted to do, there's a couple other slides on here that I just thought were really interesting. So here's a cockroach. This cockroach, you can see there's a wasp that is biting this cockroach. And so they looked at it, they said, why doesn't the cockroach die? I mean, if you take the venom out of a sting like this, it should be something that would actually kill it. A wasp, it's a lethal wasp. And so they said, why doesn't this cockroach die? And they say, it must mean that the cockroach has been exposed to this venom before. And if it's been exposed to the venom before and it doesn't die, that means it has immunity. Somehow it has a system that is allowing it to somehow sideline that venom when it comes in because they know that it's toxic. And so this cockroach has lived longer than most bugs. I had no idea. And so you would want to maybe think of a couple experiments that you could do. We haven't covered things like that, so I wouldn't ask you to do that. But it's just interesting to think that you do have organisms that may have immune systems not close to ours, but do have immune systems and perhaps do help them survive for a longer period of time. And here is one example. And another thing on here. Here was one experiment they did, but I'm not going to go through this because, well, no, I should. So when you do an experiment, and we will do, we're going to do an ELISA. So ELISA is not exactly like this, but it will be similar kind of thoughts. So here's serum from a cockroach, and you add the wasp venom to it. And if the cockroach hasn't been exposed to it, this is what's going to happen. You're not, there's no antibodies from the cockroach to be able to bind to the venom. So this will be totally clear. So now on this one, you're going to take some serum from the cockroach, and if you then give it the wasp venom, if that wasp does have some kind of response, that it has antibodies that are going to bind to the wasp molecules that are going to be infective, you'll have antibodies and they'll start to clump like this. So this is agglutination, and so this shows you that this wasp, or this cockroach actually has been making antibody or something that is going to agglutinate the venom when it comes into the system. So very similar to what we would consider an immune system. So anyway, that's an experiment that they did. Pretty simple experiment, and you can see the visual agglutination. Okay, and then the last couple wrap-up slides. Bacteria have an immune system, and we never really thought about that before. When I teach a longer course on just immunity and stuff, we always think of bacteria just really lowly little organisms, and they don't have much. But when they finally discovered that they actually have an immune system too, people really started looking at bacteria in a different light. And they found this because of CRISPR. Has most people heard of CRISPR? Okay, well, yeah, most people probably have heard of CRISPR in some way. But CRISPR is a system that's had a lot of controversy about it, but it seems like it's done a lot of good also. It's a system that you can go into a genetic material, and it's kind of like little scissors. It comes in and it binds the genetic material that you don't want in the organism, and it can clip it out. And then it carries on the other side of it, it carries the new one, and then it backs into it, and then it pushes the new sequence that should be in there to fix the broken gene. And that allows whatever organism that is malfunctioning to maybe function better again. So they're trying to use this, they've used it on organisms, insects, a lot of things, to see if this could be worked for humans. But then, there's always controversy with that. Can you really go into the genetic material of a person, a human being, and can you fix them? So they say, well, what if they have cystic fibrosis? And you could just remove that part of the gene and put a new gene in there, and they would work actually fine. And so, there's always, as science moves forward, there's pros and cons to everything. And I think that's why the more knowledge you have, the more able you are to make really good decisions when you can either say, yes, I want my money spent for that, or no, I think that kind of research shouldn't be done. But it is something, this is where they discovered CRISPR. But that's not the whole point of this. The whole point is just to say, well, what is this CRISPR stuff? So this bacteria, they have this adaptive immune system, and it's kind of like I was saying, they can chop up new sequences that come in, and they can get rid of them. So what they do is they have this, it's called cluster regulatory, you don't have to know, just call it CRISPR. So this system has a protein system, and it has Cas. You don't have to know Cas. And what it does is it binds viral DNA. It recognizes the viral DNA and says, wait a minute, I have been infected by this virus before. I remember it because I clipped a little bit of its genetic material out and I stored it in my memory over here. And so every time it's infected with something, it goes over the storage bank and it takes all the pieces of genetic material and it matches the new genetic material that comes in. And if that new genetic material has been something harmful, like a virus, it will then chop it up before it is able to integrate into that into the organism. And we're talking about bacteria, so maybe call it a pathogen. Anyway, through this, all of a sudden now this is like our immune system. What we want to do is we want to recognize something that comes in and we want to stop it before it's going to cause harm. So here's CRISPR as a nutshell. In 2020, they got a Nobel Prize for this. So here could be a bacteriophage. So this is a bacteria, here's a bacteriophage. So it secretes its genetic material inside the bacteria, and it's double-stranded, so it comes over here. And so this Cas system, it's just a protein system, it clips a little bit of this, and it stores it into its memory. So it comes down here, here's the memory, it's called the CRISPR array. So it just moves over one of these, and it puts a new little piece of DNA in. This is what I found this DNA, it just infected us, and so it clips it out, and puts it in this memory bank. And then that's it. It stays in that memory bank in essence forever. So then when a new piece of DNA comes in again, they pick some of this part up down here, and they take it over, they put it on Cas3, they hold that DNA, they come up, and they match the DNA with the new DNA that came in. And if it's a DNA that's matched what they've had before, then they know maybe this is going to be harmful to them. And what they'll do is they'll chop it up before it can actually make it into another protein or do something with it. So anyway, this is what they discovered. And my research was on virology. And I kept seeing, when I did my sequencing of my project, I kept seeing viral DNA in it. But I was really working with bacteria, and I was trying to see how viruses infect the bacteria that I was working with. And I kept seeing viral DNA inside when I did my sequencing. And so my boss kept saying, it's just contamination. You know, you have to clean up your bench. You have to, do this. You have to do that. And I said, well, I keep getting it. It's weird. So then I was talking to somebody in another lab, and they go, yeah, we do too, and we've always gotten to that. So we just figured it's, some remnant. I kept saying, why would it ever carry pieces of viral DNA inside of it? That seems like a stupid thing for a bacteria, but I just ignored it. And then I read the book about it. That's pretty much what they did. Everybody's always ignored this, and someone says, we need to look at it. There is a reason that almost all bacteria will carry viral DNA inside. And that's what they're doing. They're using it to say, oh, I matched this. This is a virus coming in. I need to chop this up. And so that's where CRISPR came from. They watched how the bacteria did this, the proteins that were used. They adapted it to being able to use for humans as like little scissors. And so this was got a Nobel Prize for somebody. So I guess I should have thought more about that, but it just it was just a very frustrating thing that you do a project and you just same dumb results all the time. Isn't it? Okay, so we are at the end. There's a lot in this this lecture and a lot in the other one. There's definitely going to be multiple choice questions on this, but there's also going to be things that say drawing or give an example of. Yes.
For our exam next week, is there going to be a study guide posted?
Is there going to be what now?
A study guide posted?
Yes, we're just talking about that, and probably the best I can do is sometime the weekend.
Okay.
Yeah. So I'd love to do it sooner, but that's probably the best I'll do. But you can, I can always say this, you can always just go through the slides yourself. It's, I could just say it's everything on the slides and leave it go at that. But I do want to try to parcel out things and also tell you how much maybe multiple choice, be prepared for drawing, whether you have to have calculations, that kind of thing to help you along. All right, anything else? Okay, so remember the food lab tomorrow, and I think you already have the pre-lab, post-lab, so we should be set with that.
Can you tell them to join?
I'm gonna be like, yo, we're gonna be studying. Love you, Vince.